is in there, and as you'll see, there's a main lab section as we walk through, and then there are the specialist rooms down the side, and we'll go into them, and David will take you through there. But this is where all of our scientists work, and so out here they can have a cup of coffee, or but in there they're gowned and gloved, and so that's the well, not only sterile, but it meets all the genetic modification, federal guidelines about handling DNA and genetic samples and things like that. And then this building is now 3 years old, and really absolutely world class. It's as good as any lab that you go into in America or in Europe. We're very proud of it. So.
Do you have any other outposts in other states?
Not labs. We have groups who support us and fundraisers in other states. We have.
Okay. Right, we're good.
Photo? Are you photo or video?
It's videoing.
Video. Okay.
Yeah.
Good to go. All right. So I'm going to hand over to David.
This is the team. So Maria, I think when have you met Maria?
No, we haven't met.
So Maria, I think after you came. So when you came last time and handed over a check, we wanted to start this whole project going. Maria was the first one who we employed to be the first person in Australia to be working on DIPG.
Thank you.
And so that's solely what she's focused on, and we'll talk a bit more. But she's made amazing progress in the last year. Both from the very beginning in terms of getting cells growing, in terms of doing tumour donations and processing that energy to get viable growing tumour cells out of that, to actually doing the first high throughput drug screen that's ever been done for DIPG anywhere in the world, and some really exciting breakthrough results that we're working on at the moment. And this is Anne and Laura, who are the rest of the team. They're actually working on other things, but their funds basically pay for them to work on other things, but also got them doing a little bit of DIPG work as well, partly because it's so exciting and they want to be part of what we're doing, and partly because there's really so much that we're discovering that we need more people to help us start to look at all the discoveries we're making. So the more funds you guys raise, the more we can do, basically.
So just as a ballpark, that $100,000 donation as contribution, as David said, that's when you take into Maria for a year plus the costs that you're doing the experimental work. That's roughly the equation, and it costs about $100,000 for a full-time scientist, and all of the chemicals and reagents that we need to do research. And when we look for good scientists, there are two things that we look for. You need to have someone who's passionate about the project, but we also speak to someone who's got good hands and can actually make, it's technically very excellent. Done extremely well with Maria.
As well as the finances question, you've got parents here whose child's tumours, they donate tumours from the children, and you actually be working on it. So, not just the fiscal side of it, but these parents have made a big sacrifice in a lot of ways.
Well, that's what I mean. You have to be passionate. I think a project like this, when you know how it's being supported and why it's being supported and what the end game is, it's easy to want to.
I think ultimately no parent who's gone through what we've gone through wants not parent to go through. So that's why research is so important.
I spoke to Chris O'Brien when he was when my daughter was ill, and I told Chris what she had, and Chris was crook at the time too, but he was stable. And I remember a pause that he said, and then he said words to the effect was, compared to our tumour, my tumour, the DIPG's, mine's a walk in the park, words to that effect. So we all know what happened to Chris O'Brien, but even at the time he was recognising the importance of DIPG research and so forth.
And so his tumour was GBM, and we've sort of always thought, well, DIPG is like a GBM. And now that these first autopsy studies have been done, we've actually found they're completely different. So in the States they did the first genetic study and they found a mutation in DIPG and they actually then went back and looked at a lot of tumour that was not found in any other type of cancer.
Doesn't
other
surprise
brain
me.
cancer,
Yeah, because
GBM.
it's so high.
It's
Yeah.
very different.
Yeah.
And so, and that's, you know, in a way, it's not, as you say, it's not a surprise because we know they don't respond like other
Shane,
cancers.
come through, it's fine.
Sorry.
And it tells us we need to be, there's no substitute. We can't be using GBM cells and saying, well, maybe DIPG cells will answer. We really need to be doing this research on DIPG if we want to do DIPG.
Penny had a really interesting question. Sorry to throw you in the deep end, but
Oh, yeah.
Wait.
So when you're looking at the DIPG tumours, do they have any characteristics that are the same? So, you know, you've got
Should we
teenage
look at
child.
DIPG?
Yeah, let's go.
yeah.
Yeah.
We go and have a look.
So what you see here in the microscope is what the actual DIPG cells look to us. We actually grow them as little spheres. So what each sphere is actually about maybe 100 or even 1,000 cells, depending on how big it is of the DIPG cells. And the reason why we grow them like that is because it's kind of the latest in science, conditions that you can grow these cells, conditions that allow the stem cells to survive. And these stem cells are the ones that actually make the DIPG more resistant to any drug or any treatment that is out there. So it took us a lot of effort, but we managed to get them growing in conditions which would allow us to maintain them under these conditions, and we have checked them many times. We routinely check them for this population of stem cells. And we've done our screens, and we have, I'd just like to show you as an example of what one of the drugs that we have tested, which is in collaboration with the level 2 adult cancer. It's a drug that we have been, it's a work in progress, but I mean, of course, what I've showed you before were the spheres of the cells, and what we can find now is that those spheres get completely disrupted and the cells die. So to go from something which is big like that to something small like this is absolutely phenomenal, and it was very exciting for us. And there was another drug that which we identified.
So what did you actually do with those cells? What did you do?
Yeah, so we grow the cells in little flasks, like these ones. And then we let the spheres to grow for about a couple of weeks. And then we add the drugs. And then we, at the end, we look at them under the microscope to see how if those spheres disrupt or if the cells stay alive. Or we find other ways or biochemical ways to determine if the cells are alive or dead. And then we sort of look at the graphs in the computer and send the reports to our boss, David. And then boss will and then David tells us, yes, you do it again. But yeah, I mean, yeah, what I've showed you before is that, yeah, we've seen really dramatic changes with certain drugs, and we would like to take this further. How long does it take from here to actually get those steps for the kids?
Oh, so well, it depends what drugs they are. So yeah, so part of what we've done in the screen, we've screened about over 3,000
compounds,
over 3,000 drugs, and the vast majority do absolutely nothing. But what we
Not
found,
surprised.
yeah, not surprisingly. So you look.
A lot of them have been tried in kids already.
Yeah, exactly. So, and of course, you can't take 3,000 drugs and test, all of those in kids. So this is an amazingly fast way to just rule out thousands of them. And out of those, there's about 50 that seem to be active. And so now we're in the process of narrowing that down further, because some of them don't get into the brain, so they don't get around the blood-brain barrier. Some of the delivery, some of it we need to repeat it in other cell lines that we have, like we've got more than one DIPG cell growing to make sure it wasn't just the one we tested, it actually works more widely against others. We need to test them against sort of normal healthy cells and make sure they're just not killing everything. They'll be too toxic to take. And then ultimately we want to test it in sort of animal models. Before we give it to kids. And then depending which ones are the most active will determine how quickly we can get that into the clinical trial. But the 3,000 we've started on, obviously the easiest ones, the drugs that we already know about, that have been approved many times, kids have already had them, so that anything we find there, we hope in the next couple of years we can actually start treating.
So that's just as an example of that, Project David and I have been working on the neuroblastoma, which is a tumour of the nervous system, not in the brain, but down in the abdomen usually. We found results in cell culture and then in animal models back in 2008 that suggested that we had drugs like this, approved drugs, the same sorts of drugs that the guys are currently screening. And we found that a particular set of drugs seemed to be working very well on very resistant, very refractory neuroblastoma. And we presented that at international meetings in 2008. We've just been there's been a very intensive period of validation, testing in other models, confirming in other laboratories, making sure that it wasn't just one line, repeating it several times. But in the space of just on 4 years now, we've got a clinical trial, we've got a teleconference on Friday, the drugs are synthesised, ready to go, the hospitals are lined up, the new drug protocol has just been approved at federal level, and that trial should open in the next couple of months. So in a 4-year window from the time that we demonstrate the first results to the time that you're actually into the clinic, can, if we're lucky with the drugs that you come up with, be something that is actually happening.
And Michelle, what new avenues or methodology would there be to try and infiltrate the blood-brain barrier with DIPG, which is what, as you know, sets it apart from so many things? Are there new techniques or methods that you could consider or are worth following?
Well, the initial thing we're doing is just actually choosing the drugs that we know get through by themselves. So we don't have to try fancy, difficult things to do, but that's the simplest, that's the fastest way to get it through. You choose the ones that it's going to be most likely.
Because the ones that get in.
So but get into the blood-brain barrier. So if it's the right size and shape and dissolves through, then that's perfect.
So that'd be the best case scenario. That that one amongst this short list of drugs that work, there are those that really cross the blood-brain barrier very effectively, and not just when tested in one cell line, but in 2 or 3, and then in the mouse models, make it through each of those hurdles. If you got one of those, that would be just about the best we could hope for.
And would you say, Michelle, if say best case scenario, and you had success in time with the various drugs and protocols, would you say that successful DIPG treatment could provide a template for other cancers or successful outcomes in other areas?
Well, I think yes and no. I think, like I talked about earlier, when you look at the genetic changes in DIPG, they're unique. So that's why we need to focus on DIPG itself. If we're going to cure it, we need to find what works with DIPG. But yeah, it's possible, particularly when you look at things like combinations, that something that works in DIPG may work in other things as well.
The same thing with DIPG too is that I found that a lot of them, the makeup is different in every single trial.
Yeah, well, that's part of the challenge too. There's going to be what we call heterogeneity. So there's always differences. There's different genes switched on and off. So
what you sometimes find though is that there are critical genetic lesions. The tumours have lots of genetic changes. That's part of the hallmark. We call it the genetic instability, normal genomes in normal people, the normal makeup of the DNA is extremely stable. It replicates from one generation to the next throughout life. And each cell looks like the daughters and the granddaughters and the great-granddaughters, whereas cancer cells are known for very rapidly changing mutations. And some of them are what we call driver mutations, the ones that are actually responsible for the cancerous behaviour. So this genetic analysis of the DIPGs is really just starting. I mean, you know, in this last 12-month period, we're only just now, with the benefit of these autopsy samples, beginning to know what the genetics of this are like. And what we have to work out is, amongst that mess of changes that are different in each of them, are there common ones and are they the critical ones? And if you can find them and targeting them, then there's the answer to your question, well, are there other tumours that have similar mutations?
Yep.
And if they do, then the treatment that you find for this is likely to also be effective in that. And an example of that is that there's a mutation that's found in breast cancer quite commonly that's responsible for familial breast cancer. It's also responsible for familial ovarian cancer and also pancreatic cancer. And it's the same mutation in all of those, and the drug treatment that they found that targets that mutation that's in clinical trial in breast cancer and ovarian is now being translated over to pancreatic. So it's like a jigsaw puzzle, and you sort of sift your way through the data and begin to try and find out. So this is the beginning of the journey, but it's a journey that hasn't started.
So would biopsy be more of an option then? You know, I know they don't biopsy DIPG,
Well,
but if you're heading down this path...
Yeah, so there's actually a protocol open in the States now where they're doing that. So they're doing a biopsy when it's the first time diagnosed. And yeah, I mean, depending on what we find...
Because in Australia, we were told no, we don't do it.
Yeah, so it's experimental, so that's what they're trying. But yeah, it may end up that we end up going down that path.
Because one is going to be different to another.
Exactly, these tumours will respond to this drug and these ones won't. So.
Yeah, and that's in this example that I spoke about, the breast and ovarian cancers that have this mutation go on that drug, but the ones that don't have to have a different type of therapy. That's exactly right. And you see, the reason that we don't biopsy is because there was no point. Why biopsy? What were you going to do with the information? There was nobody doing any research, so we didn't know what to treat them. So when I say this is the beginning of the journey, it's both from the genetics, which is happening overseas, and from the therapeutics, which is now beginning to happen here. You combine those two together, and that's how you make the quantum advances. And
a year ago, no one had ever grown DIPG cells in Australia.
Yeah.
So what you're seeing now, you know, is the first time these cells have ever been grown.
They did first. I did in,
I'm not sure where first.
there was a mouse model in the States.
Oren Becker.
Yeah.
Well, that's where this whole thing started. I heard Oren Becker speak at a cancer meeting in 2011 in Washington, and I came running home from David and I said that the DIPG mouse model.
He's a very unassuming man, but he's
lovely.
And he works in conjunction with us as well, you know, and I'm sure that Oren will speak highly of our organisation, as with Miriam Laudy working with Tim, you know, so. And we're all about collaboration. I think I've tried to reiterate that with the, you know, and that was a big part of the funding. That was, you know, it's imperative that CCIA have open arms with institutions Australia-wide, and that's what we're all about, you know, because we deal
at all levels, you know, to collect the samples. It needs to be national. To do the trials, it needs to be properly international.
Because again, need to be the focus, and trust me.
Another thing that sets our illness apart is that by the time a protocol may be seen worthy or fit to trial, fair odds are that the child will be dead to even try it. You know? So we have a fair amount of urgency in our work, if it hasn't come through.
Not to be pushy.
You know, there are times, I think there must have been times when for any DIPG therapy, even now it's still almost a hopeless cause. But if you think back, it's only 50 years since that was the case for acute lymphoblastic leukemia. Every child with acute lymphoblastic leukemia was dead within 8 weeks in 1960 when I was a child. And now 85% of those kids are going on to long, happy, healthy lives. That's 50 years, it's not thousands of years. And it's all through the sort of stuff that we're doing here.
And you just think as a DIPG parent makes you upset because in 50 years, DIPG just hasn't even been acknowledged. you know, so
So
just
you're
imagine
behind
if
the
that
eight
had
ball, absolutely.
Yeah, you just imagine if this had happened back then, maybe my daughter would still be.
I know.
That's the urgency that we kind of talk about more so. That we come a long way with breast cancer, we come a long way with leukemia, but this whole level of there's only a few of them, so we'll just let them kids die. So we'll just let those 20 kids in Australia just die every year, because that's basically what it is. It's a death sentence.
So that's why I love the name of your organisation, The Cure Starts Now. I mean, it actually does.
And it was named for a positive connotation too, Michelle. You know, we didn't name it after a particular child or a particular a flag for one particular family or thing. You know, we named it for a positive connotations, which it does. You know, and as you've acknowledged, we are starting from scratch, so it is a big thing. Just a quick one, Michelle. You mentioned like driver cells or cells that. Do they look differently when you're looking at the raw tumour cells? Do those actual cells look different?
Defer that to the guys who are in
No,
the
they all
just
look very similar. So you can't really say from a microscope that they might have something different to them. So they all look very, very similar. They're all like spherical tumor cells. So
with the different tumours, how many types of cancerous cells are there in the tumours? Like, is there 3, 4, 1, 2?
They're all cancer cells. So what we have in here, they're all.
But are they different types of cancerous cells, or?
Yeah. So what I think what we're talking about is the stem cells versus the non-stem. So they're all cancerous cells, but we think part of them are the physical, like the driver cells, the ones that you need to eradicate if you're going to get rid of the tumour. So part of they don't look different; they look exactly the same. Part of the challenge is to find the markers on those cells, and then sort of split them off and say, well, whatever new treatment you have, is it actually directly attacking those cells? Because if you're not getting rid of them, then you're not going to get rid of the tumour.
Do you try combinations as well, or are you just doing one?
It's absolutely combination. So part of it is sort of thinking, well, if this works, maybe we'll add this drug. But what we're also planning to do is another screen actually in combinations. And that's been really tricky actually to work out how you do that, because you said, well, we did 3,000, but if you took 3,000 drugs and combine that with the same 3,000, that comes up to about 9 billion. Yeah.
3,000 by 2,999 by 2,999.
It comes to 9 million or something.
A fair bit.
Combinations, which it costs $2 for each screen. So that's, you guys would need to raise $18 million. So, but we're working out different ways of doing that. Actually, just maybe taking 100 drugs that are most likely active and combining that in those ways, or taking groups of drugs and saying, well, this one represents this 30, and then work out which groups of drugs and all that. So so that's one thing.
But without being a little bit facetious, and I am a little bit biased, and I make no mistake, like say that if there was a set figure of $18 million and you could even partially assure an outcome compared to the funds that other larger cancer groups spend, like for example the research being undertaken by the mainstream cancer groups to help reduce nausea to do with chemotherapy intake and so forth is millions of dollars in Australia. So as it stands at the moment, we're spending more money on trying to stop people from vomiting than we are trying to address this. So we are a little bit biased.
Yeah. Well, certainly the more.
So I'll quote you on the 18 million.
Well, if you raise 18 million, I might have to work out what's the best way to spend it. But the more we raise, the more we can do. Like each of these things that we're finding in these screens, is a path we want to follow. So,
and that's why guys
somewhere in there we know there is a cure. So.
And if everyone here can implore in their dialogue with the professionals and their colleagues, just mention those four letters, DIPG, because until we get it recognised in circles, we'd really love to get that 18 million.
In the meantime, until we have that, as David said, the strategies to, when you say if there was even a reasonable likelihood that it would work, you can design the study in a way that you'll find out pretty quickly if it does have a reasonable likelihood that it will work. You pick your best champions and see, and if when you combine them with something that's working really well, you get something incremental, then you're on the right track, and then you can begin to hone it down. So you don't really need to do just a random, combine everything with everything. There are smarter and faster ways.
Yep, yep, I know.
Right. Should we,
No, we have to take those cells back to the incubator.
oh yeah, keep them,
They get cold.
look after
They actually do get
Yeah,
cold.
treat them very gently.
Yeah.
Alright, shall we head back upstairs?
Yeah.
Or-
Do you guys remember the tumour bank? You've seen that before where we store the tumours. Okay, so we've actually got here where the tumours go into liquid nitrogen and we can keep them from the
I think I
time.
saw that on the YouTube video from last time.
Right. Okay, good.
Is it possible to do a drive-by the tumour bank?
We've already got a YouTube video of
Oh, have
So
you?
if you
Okay.
want to pop in there for 2 or 3 minutes, we
Sure,
can. It's
I wouldn't mind.
just down the corridor. Thanks, guys. Thank you, Marie.
You're
Thank
welcome.
you.
I'll see you soon.
Thank you, girls.
Should we have a shell? This is the tumor bank.
Very naughty. We have to.
So this is one of our incubators. You can see that it's kept it as cold as possible, essentially, minus, what is the temperature? Minus 163 degrees.
So at the bottom of that was actually liquid nitrogen that's been pumped in along these tubes, and the temperature of liquid nitrogen is minus 196. But we keep the cells in what we call the vapor phase in the gas, and that's at 165.
So I'm not sure we've got to this particular bag. Basically, each of these racks. Box.
I'll just do it quickly so that a floor of it inside of it, each of them is a 10 by 10 rack, and that's each of those is a sample of tumour or
Of the same
living
tumour.
cells. They're all
Well, it's
alive.
everything.
This may not be a DIPG rack,
Oh,
but,
yep.
but each of those is a sample from one pure tumour or cell from a particular patient. And so we can keep them, and they're alive. They're just in suspended animation, like in science fiction. And then we can pull them out and grow them and turn them into those cells growing like that as they were in the laboratory.
So when you're doing like with an autopsy donation, say it's done in a country hospital or whatever, do they have to be transported by dry ice, or are there obviously certain protocols with
there's-
that? Or?
Couple of different things with, so there's a whole, it's a whole complex protocol that we do. So there's a few different things we're trying to collect as much as possible. So part of it is the cells we want to grow. Actually, we're transporting at room temperature and then basically starting to grow them immediately. So to grow them, they need to be at 37 degrees of body temperature. So that's part of it. Other bits we're actually freezing down and keeping for later genetic analysis, etc. Collecting a bit of normal brain tissue as well, so we can do comparisons. You know, if we find something particularly interesting, we want to know, is this actually just part of the tumour, or any normal brain to compare it to?
So they're not one lump sum, just all frozen,
No, you can divide them.
So we divide it into the different bits that we need.
Have you had much luck with tumor donation in Australia?
So we've done 2 so far.
Since bringing the protocol in?
Sorry?
Since bringing the protocol in?
Yeah. So, and it's just starting to open.
When was that? When was the 2 donations?
Sorry?
When was the 2 donations?
When? Oh, in the last few months.
Yes,
So
one of them would have been my son.
One of them was Talin, who was the first, and then one other since then.
So,
It's very, very good curves.
it's a bit of
Bittersweet.
bittersweet because because obviously we don't want to have any child to have to donate their tumour. But by the same token, to have that opportunity to do that research, to stop, to make a difference for children in the future, that's what I'm talking
And I think that's the whole point. You know, we made promises to Liliana. We said to her, you're not going to have this tumour anymore. So you have to keep that promise. You know, how do you tell a child, no, you're not going to have it anymore, but then bury that child with that tumour?
Yeah.
You know, so that's what kept us kind of a little bit sane about the whole tumour donation process. And then also knowing that hopefully one day someone didn't have to go through what we went through and didn't have to experience what she had to experience. We will get there.
Yes.
We really will get there. That's what gets us out of it. David on the clinical and the research side, and me on the research. This partnership between the clinicians and the researchers is actually a tried and true formula, and when it works well, it leads to cures, and it's as simple as that. You need great science, you need committed people, and you need funding. And when you've got those three, you'll find the answers. Oh, I mean, I absolutely believe that.
Yeah, for sure.
Absolutely.
Thank you. That's good.
Complex issues.
Yes.
They're very complex issues. If they were easy, Ren, we'd have solved them a long time ago.
Yes.
So it's really interesting, but something we need to think is unlikely to be the
And mice are the preferred tool, obviously, or they
I mean, what was it, just 18 months ago? Last time you were here.
There you go. I was trying to work it out before.
I think it was 18 months, and I hope that you can see what's happening as a result of the funds that have been raised from a time that you were saying to us, can you do something, can you make a difference with the benefit of the funds that you've given us. I hope you will leave here with a sense that what's actually happening is something that has a genuine capacity to make a difference, that this is really, as I said, the beginning of a journey that has, I mean, it's not just a light at the end of the tunnel, it's a shining light. If we can get the outcomes that collectively we all want, the guys who are doing it, and David who's actually, you know, treating the kids as well as doing the science, and you guys who, you know, had to go through this together, what you're doing is finding that path and lighting that path, and with that we can hopefully move along that path towards where we all need to get to. So it's a wonderful partnership. For us, this is our mission. This is why CCIA was set up. It was to have a group of parents who, it was set up by parents who wanted to make a difference because of what had happened to their kids. That is exactly from where we came. And in the entire time I've been here now for 28 years, that mission, that vision hasn't changed. And over that time, the survival rates for many of the cancers have improved dramatically. Where there is one like this, when David first started talking to me about DIPG, and I said, oh my God, you know, I actually hadn't really understood the extent of the challenge until he said to me, Michelle, nothing. And it was really, Ren, it was your initial case and your early conversations that focused me on this, even in an Institute of Children's Cancer, that suddenly made us realise that A, this was something that we had to do, and B, that we could do something with the technology that we've got, and then C, with the support of the community. That was the final ingredient to make it all happen. So we're humbled by your support. We know why you do this, and we see your pain, and we want to one day change that so that other people don't have to go through that. And so for us, it's just a privilege and an honour and a pleasure to work on this. And it is our hope that with the support that you give us, we will make a tangible difference to children, to lives, to families, to parents, and that we will one day do with this disease what's happened with so many of the other children's cancers. So I just want to say thank you to you guys for what you do. I want to tell you that we passionately believe that we can do something to make this better, and that it is a partnership, and that as long as you will continue to support us, we will give heart and soul 150% to try and make a huge difference going forward. So that's all I wanted to say. Thank you.
No, thank you very much.
And Rem, I'm sure.
Oh, no, it's just a privilege for us to be here. You're probably not going to strike too many more informal organisations than ours, because
we love it. We're good at that.
It's just the way it is. It's just the way it is. But guys, we just hope to. We hope that one day we can get the silver bullet of a successful treatment into a phase 2, 3, 4 trial.
Where's getting the kids?
Yeah, we've got to get these things, because those of us intimately involved in our organisation, you know, you can sugarcoat things, but we deal with death, you know, and it's an ugly thing. So what motivates us is trying to prevent this and get this story out to a wider audience. And it's actually a travesty that it's not more widely publicised, but that's just my little bugbear. Guys, everyone in this room who's concerned with DIPG, have a look. I implore this, because you'll see some things happening over the coming 6 months on the DIPG registry, DIPG.org, the registry that our organisation contributed $25,000 to help get established, and we're working with Marion Falloudi and Tim Hasseldet to get to create specific benchmarks and all the things that you guys deal with, all the autopsy donation protocols, get them all registered, and I'm sure you guys will work with Tim a little bit more. And again, thank you. We just need to get, just out of interest, if you did find a successful agent or whatever, how long is there would it take 6, 12 months, 2 years, like to get it into a trial? Like is it I dare say it would be logistically tricky.
Yeah, so it really depends what it is, and you know, a lot of the time, you know, first thing is to do the science, and with the science work out what's the right drug, what's it doing, does it actually work, and then the next part after that is more the practicalities, and that often comes down to who owns the drug, who manufactures it, you know, how much does it cost to make it, is it something that's available, or is there a similar drug that we can use, you know, and that's really what it comes down to. So, you know, we've had drugs that, you know, like Michelle was talking about, a drug in another tumour, neuroblastoma, that was a very old drug, but no one was making it anymore. So to get that into the trial, we actually had to go through the whole process of finding a drug company and get the whole process going to actually pay them $350,000 and actually make the drug from scratch. There's another trial that we're doing in leukemia where there's a drug company that's making it, and they're very keen, and they're going to, you know, pay for all those costs, and you know, within a year of going to them, we can have the trial up and running. So it really just depends what we find, but you know, the strategy is to start with the lowest hanging fruit, so the things that are going to be the easiest and the fastest to get to the clinic. Ultimately, you know, we want to look even broader and discover new treatments, and you know, things that may not even be drugs yet, and that might take, you know, 10 years to get it from the point where you've got a chemical to a drug that you can actually give to people or to children. But I think the key is to try and attack it from as many different angles and as many different options, and if one falls through, or you've got something else, you know, that's coming up as the next in line, or if that doesn't work, looking at the combination, you know, how we can improve it even further. So, yeah.
So no black or white.
The simple answer is there is no simple answer, but our certainly the way we work is to do it, you know, to look at the practicalities and try and get it done as quickly as possible. And certainly my focus is always the aim is translation. The aim is getting it to the clinic. So the aim is not to, you know, have a lab of 20 people doing more and more.
Right, now, knowing what you know, would you be more willing to try things off trial?
Oh, I don't think I've got anything yet to say that this is what I, you know, go to a kid tomorrow and say, yeah, this we should try this. But you know,
there's a lot of parents, because in Australia, especially because there are no open trials as such. You know, off trials are always an option, and knowing certain aspects of drugs that you now know of. Would you be recommending to those parents to try off trial?
So I don't think
drugs?
we're at that point yet where we could even say, oh, this, you know, looks really good. You know, we should take it even though we don't have a trial like that. But ultimately, what we really want to do is make sure those trials are available.
Oh, obviously, I don't just because we did something off trial. So because obviously in Australia there is nothing. There are no trials. There were no trials. So you know, you're weighing up your options, and that's just knowing what you know. I just didn't know what that was. But the focus is, as I was saying, not just to do the lab research and fill journals with articles, and you know, the aim is actually to get to the clinic when kids need it.
And when I was saying downstairs in the Biobank room about partnership between clinicians and scientists, that's what makes CCIA so unique and so valuable. And what we're proudest of is that through the links that we built with the clinicians, and in the best case scenario, actually having the clinicians embedded here in the lab as well as at the hospital, like David, it means that where we do find treatments that work in the lab, we don't have to then create a pathway to get them into the clinic. I mean, that's David, part of his role is to develop clinical trials for new protocols. So it's unique actually having David running this project, because he's probably the leading person in the country with clinical trials experience for kids' cancer, and also focusing on DIPG. So in terms of having the infrastructure in place to develop new treatments, what we've got here, this partnership is actually the perfect situation to go from, as we say, bench to bedside, laboratory to clinic, from concept to testing straight through into the clinic. So the only answer we can give you, Ren, is that if there is a path that begins to become apparent that this should be going into children, we will do it as fast and as effectively as anyone in the world.
Yeah. So if you found one, say tomorrow, like is there a to do with all the legalities and ethical things? Does it take 6 months, 3 months? Just out of curiosity, I have no idea.
So if we had something tomorrow that said if we did all the lab work, we had everything saying this is the drug that we want to try. Realistically, to get it to the point where we're treating the first patient, from that point it's probably about a year. Yeah. Just out of interest.
But not 10 years.
Not
5 years.
No.
I mean, this one that we've just done, as I said, was 4 years, but 2 of those was fighting with a bloody drug company to get the drug synthesised, because they hadn't made it, and then they had to make a special batch, and then they wanted top dollar, and I mean, it really nearly stalled over that, and it was going to be over literally my dead body. I wasn't going to let that happen, so we pushed it through, but that just delayed the process by 2 years. But had funding not been an option in that situation, we would have been there probably 18 months more quickly, I reckon.
It's terrible that drug companies can actually stop children getting this treatment because
It wasn't that they wanted to stop it, they just said, look, it wasn't a big pharma, it was a small, newly developing company that just was literally said, initially they said they were going to fund it, and then business went bad and they said they didn't have the resources, and we just had to find the funding. And then when we got a source of funding, then they were very good. You know, there are often these unforeseen obstacles, but part of our job is to clear those obstacles away and to sweep the path, and the more often we do this, the better
And
we'll
to
get
sit
at
down
it.
with the drug companies and say, well, sometimes they're concerned, they don't want to give it to children because they're worried the children will get side effects, or so we go through that with them and explain that's not an issue.
No. It's a choice between side effects, and the inevitable side effects would be.
Yeah. Well, that's all I expect.
Thank you.
It's just kids, we're all given some different friends and things.
Oh, right.
Fantastic, thank you.
Bits of our children in there.
Yes.
Favourite family recipes?
Any particular one you recommend from here?
They're all good.
Maria can probably give you a recipe for growing tumour cells.
Quite
Not
the same.
No. So you got 4 more to go. Oh, no. Cheers, cool.
Thank you for that, that's great.
Encourage this to cook. So easy. That's what my character.
So you want to go around and hand over before I run out of battery?
This skin slicing is really nice.
Yeah.
Did you actually have a big check? Would you actually have one? We have everything gone up in the world. We don't have an oversized one. Sorry, here we go.
I'll just keep this one.
There we go. You, but for publicity purposes, it's thank you so much.
Thank you, Mr.
No, that's okay.
I'm not sure where to look.
I'm just going to get another camera. Oh.